Good evening and welcome everyone to Duquesne University's 2021 Darwin Day celebration, capped off by a talk tonight by Dr. Michael Warby of the University of Arizona. My name is Michael Seaman. I'm an associate professor here at Duquesne University in the Department of Biological Sciences. It's my pleasure to welcome those of you who are joining us virtually tonight. I'm sure we have a lot of students, faculty, and staff coming to us from Duquesne University. I also know we have a lot of others coming from other colleges and universities in the area. So I want to give a special welcome to all those in our general neighborhood here, especially the students and faculty from CCAC, University of Pittsburgh, Penn State, St. Vincent, Seton Hall, Teal, Clarion, Slippery Rock, and everybody else. I know Mount Aloysius and Moravian is in the audience, so welcome to all of you. But we should remember that regardless of where you're coming from, we all got here the same way. And I don't mean a Zoom link. We all got here via 3.5 billion years of mutation, natural selection, and other evolutionary forces. So congratulations, you have evolved. Happy Darwin Day. Before I introduce our speaker, I have a few points of business. First of all, there will be time for questions and answers after the talk when Dr. Warby has finished speaking. Well, if you have questions at any time during or right after the talk, please add them in the chat feature, which might be labeled as Q&A or chat in your Zoom link. And I will moderate those questions and ask those of Dr. Warby. So don't be shy. Secondly, a reminder that anything and everything related to Darwin Day at Duquesne University can always be found at the easy to remember URL www.duke.edu/Darwin. There are links to past events. We have the recordings from the last 10 or 12 years up there, as well as notifications of future events. And tonight's talk will also be recorded, and within a day or two will be available at that link. So if you have friends and family who couldn't be here tonight, be sure to let them know, and you can find the talk there in a couple of days. Well, it seems like everything with the pandemic over the last 12 months has gone virtual. And Charles Darwin is no exception. With this award-winning interactive app developed by Drs. John Pollock and Dave Lampe of the biology department here at Duquesne University, you can have a real conversation with Charles Darwin, or virtual Charles Darwin. You can ask him questions because he has answers. So check it out, www.thepartnershipineducation.com. It is available on whatever your favorite app platform is. Please check it out. All right, let's get to the reason we're here. We are here to come together to celebrate what would be the eve of Charles Darwin's 212th birthday, born in February of 1809. We're here to reflect on the life and work of Darwin, the founding father of evolutionary biology, and really of all of biology, because evolution is the single unifying principle that brings together all of our diverse fields of study. I know it's been said a million times, but I'll say it again. Nothing makes sense except in the light of evolution. We also use this as an opportunity to celebrate the importance of science education. That's something very near and dear to us here at Duquesne University. So it is in this vein that I extend a special welcome to those in the audience who are not professional scientists or educators or students. Welcome, and we're glad to have you here. We're proud to make all of our Darwin Day events available to everyone, which is motivated by Darwin himself. I know he wrote a lot of articles in obscure scientific journals like we all do, but he also wrote popular books to explain the wonders of the natural world to all people. And he gave many public lectures to explain his findings and his theories. Over the last 20 years, we've been fortunate to have the support of the Baer School of Natural and Environmental Sciences and of Duquesne University to bring in so many truly outstanding speakers over the years, including Sean Carroll, Peter Ward, Marlene Zook, Richard Lenski, Rosemary and Peter Grant, Hopi Hoekstra, and May Berenbaum, just to name a few. But these things don't organize themselves, so I would like to quickly thank Dean Phil Reeder, the Dean of the Baer School at Duquesne University, for financial sponsorship and other help, Ms. Walters, Ms. Stone, and Dr. Palmer in the Dean's office for logistical support. Thank you to Dr. David Lampe, my co-coordinator for Darwin Day now for well over a decade. And I want to thank all my many comrades across the university for their help, including catering and event services, graphics design, web hosting, video recording, emailing, and other support. So thank you for your help. It's now my pleasure to introduce Duquesne University's 2021 Darwin Day speaker, Dr. Michael Worobey, the Louise Farquhar Marshall Science Research Professor and Chair of the Department of Ecology and Evolutionary Biology at the University of Arizona. Please join me in welcoming Dr. Michael Worobey.
Hi, everybody. Thank you very much for that warm welcome. It's really great to be here. Thank you to Michael and Dave for inviting me to be the speaker this year. It's a real honor and privilege to be here. So I am going to share my screen now and get right into it. And we've already seen a picture of Darwin from when he was a young man. And one of my favorite little bits of scientific notation is this page out of Darwin's notebook from 1837, where he says, "I think," and then draws what is probably the first phylogeny that anyone ever put on paper. This idea that organisms are related by descent from common ancestors. And this has become a very powerful metaphor, a very powerful tool for understanding biology, and in particular for understanding pandemics like the one that we're in the midst of right now. And so we're going to fast forward now 184 years or so and get right down to what's been happening here in the last year. So on January 21st, 2020, last year, the news broke that the first COVID patient had been diagnosed on US soil. And this was a very interesting story, not just because it was the first patient, but it was also a case where the response by public health authorities and by the person who was infected was really exemplary, and all the right moves were made. This person was a Chinese national. He had gone back to China, to Wuhan, to visit family. And he was aware of CDC messaging about this new pneumonia illness in Wuhan that was causing a lot of illness there. And when he got back to SeaTac, January 15th, a couple days after that, he started feeling a little under the weather and thought, well, I better go to the doctor and talk about this. And was able to get a diagnosis. He got the dreaded nasopharyngeal swab, and that swab sample was sent to the CDC. And sure enough, this guy became the very first case on US soil. He also became the first patient to have a complete genome sequenced. And this has turned out to be an extremely powerful tool for understanding when, where, and how this epidemic originated, spread, and now how it's adapting to the human population. And it's basically what I do. We sometimes call it genomic epidemiology, because we use these genomes and these family trees, phylogenies, to use the fancy term for an evolutionary tree, to try to answer these sorts of questions. And actually, just while we're still on this slide, one of the first connections to this story for me was chatting with a grad student in my own department here in Tucson, Arizona. This would be in February. This was in the before times when we still were having receptions and not wearing masks and all of that. And he mentioned that he had been on the same flight as this case, and had been contacted by public health authorities wanting to know if he had been sick. And it was just a good indication of how well that first response unfolded to try to basically prevent onward transmission from this case, which has been the sort of thing that we've been trying somewhat unsuccessfully to do in this country since January. Okay, so now fast forward 6 weeks later, and we have another story breaking. Coronavirus may have spread in US for weeks, gene sequencing suggests. So this refers to a finding by Trevor Bedford and others involved in what's called the Seattle Flu Project, where researchers had been taking samples from people's noses for some months prior to the pandemic. And the idea was that they were going to look for influenza and other respiratory diseases. And it turned out to be this kind of gold mine of samples that you could look for the virus that causes COVID-19, SARS-CoV-2, as we call it. The problem was the FDA essentially said, cease and desist, that you're not set up for diagnosing people with this illness, so don't look at those samples. And finally, after 6 weeks, they kind of threw their hands up in the air and said, screw it, we're going to look at these samples and we're going to see if any of these people that we sampled in January and February actually have this virus. And sure enough, a case from late February became what's called WA2. So the second case in Washington state, the first WA1 being the Chinese national who had flown into SeaTac on January 15th. And now this is where things started getting interesting from an evolutionary perspective. And this is from Trevor's paper in Science. So what we're looking at here is an evolutionary tree of the virus where every step between those dots is one mutation. And over on the left, you have the sort of common ancestor genomes. Most of these were sampled in China. So these darker blue lineages are from Asia. So most of these are samples from China. And then as you can see over time, this virus, although it started as a single infection in one patient back in China, has picked up mutations. And this allows us to make a family tree of the variants of the virus. And in red, you can see samples that were sequenced as part of this Seattle Flu Project in Washington state. There's a minor lineage here with a small arrow, but most of the samples from Washington state fall in this big grouping here that we call a clade. And this was very interesting. This indicates that the vast majority of people in Washington state over the spring period actually had a virus that descended from a single introduction. And things get even more interesting when you look at this clade. If you zoom in on this clade and the most closely related genomes in the data set, you can see this WA1 sequence. It's in red here. It's kind of hidden by some of these Chinese genomes. But this WA1 genome is actually only 2 mutations different than virtually all of these Washington state outbreak viruses. This is 2 mutations out of 30,000 nucleotides in the genome. And if you go back and look at this phylogeny here, if you think about how this Washington outbreak clade got established, the chances are actually kind of slim that this would have been a separate introduction of the virus, because if it was a separate introduction from WA1, it could have landed over here or down at the bottom. It could have been a very different virus that emerged. And these researchers concluded that most likely what we're looking at here is a situation where WA1, despite all of these efforts that were made to prevent onward transmission, right down to a sort of a biosafety level 4 suit that he was put in when he was moved into the hospital. And despite all of the contact tracing with people that he may have contacted to see if people had become ill, the conclusion was that this individual probably had started the outbreak in Washington state because the viruses are so similar. And that had some pretty big consequences and was a huge wake-up call for the whole country and Washington state in particular, because it meant rather than being at the start of this outbreak of COVID-19, that Washington state may already have been 6 weeks in at that point. And if that was the case, there were probably thousands of people infected and so forth. It was a real sort of emergency finding. And at first, when I saw this finding and followed the logic of the similarity between the outbreak clade and WA1, I was pretty convinced that this was exactly what had happened. But as time went by and I started analyzing these gene sequences, I got more and more used to the sort of pace at which this virus evolves. And it started to strike me as a bit odd if indeed WA1 had started this outbreak, that, for example, we weren't finding any genomes that were identical to WA1. All of them had these two diagnostic mutations separating them from WA1. And in Washington state, we also didn't see any viruses that were only one of these two mutations away. All of them had these two mutations. And that led me to a sort of quest to really try to test in a quantitative fashion this hypothesis. Did WA1, did this patient on January 15th really initiate the US COVID epidemic? So this work was eventually published in Science magazine, and it involved the story I'm going to talk about today, and also another one that I won't touch on, but maybe we can talk about in the question time, a sort of similar situation in Europe, where there was speculation that the large Italian outbreak in northern Italy had been started not by a virus moving directly from China to Italy, but from an earlier movement of the virus from China to Germany, and then from Germany down to Italy. Okay, so what did we do here? What we wanted to do really was rerun the tape of evolution over and over again, under the constraint that in our computers, we can specify when we go in and play God and simulate these epidemics, we can specify, okay, that this virus really, this outbreak really did start with this January 15th patient. And let me just take you through some of the logic here. If you look at panel D in this figure, this is a representation. Each dot is a person. And so over on the left-hand side, you might have an individual becoming the first to be infected in this transmission network. Over time, in the next unit of time, that individual has infected two others. And as time goes on, infected individuals pass the virus on to other people in the transmission network. And whenever there's a color change here from this yellowish color to this maroon color, for example, that's indicating that as well as moving from person to person, the virus has picked up a mutation. So something like these two diagnostic mutations that we saw on the previous slide. And so if you go to the right-hand side, this is what you end up with when you sample the sequences from Washington State in reality. All of them are 2 mutations away from WA1, and there's nothing in between. So that's one possible thing that you could observe if WA1 had really started the outbreak. But there's a whole bunch of other things that you could also observe. So for example, in panel B here, we have a similar transmission network, and we get an outbreak clade, these blue sequences that look like the ones that we actually observe. But in this case, WA1's genome has been passed successfully to the present time, and so it ends up getting sampled, and you get this simulated phylogeny that has genomes that are 2 mutations away, but also you're sampling some genomes that are identical to WA1. And these other panels show other possible scenarios where you could have a variant that shares one of the 2 diagnostic mutations, or none of them, but has accumulated a separate mutation down a different pathway than the actual observed sequences. And so the question is, how surprised should we be to see this pattern where you only see genomes that are 2 mutations away from WA1, if that's really how this virus got its start in the US? And so here's where you go into your computer and you rerun both the tape of the epidemic unfolding in Washington State and the tape of the evolution of the virus during that unfolding of the epidemic. And so we use quite realistic parameters to set up a transmission network of individuals for the virus to go from infected person to uninfected person and move through that population. And we just allow that to happen over and over and over again, where you start with one patient and you have the epidemic spread through the population, and then you evolve the virus starting with WA1's genome. And again, this allows us, it doesn't mean that we're sure that that's how it started. It means that this gives us a very powerful way of asking, if that was how it started, what evolutionary patterns would we expect to see? And so here is what we would expect to see. Let me just draw your attention to panel A to start with. In panel A, this is essentially what we observe in real life, where you have WA1 and then you have 2 mutations separating WA1 from everything. That's actually something that's kind of uncommon to see in the simulations. If you look down at the bottom at panel E, 100% of the simulations, when you have outbreak clade sequences that are 2 mutations away from WA1, as these ones with an O are, 100% of the time, if you see that pattern, you should also expect to see what we call a sibling lineage, where it's a lineage that shares one of the 2 diagnostic mutations, but then it's picked up some other mutations along the way. And in fact, we don't see those in Washington State. And that simulation study then gives us pretty strong evidence that this virus, this outbreak probably didn't start with this patient WA1. The patterns, the evolutionary patterns that we see are just not consistent with that. And so that's one way you can get at this. The other way to get at this takes us back to that figure of Darwin's phylogeny at the beginning of today's seminar. And this is, in a way, an even more powerful way of asking this question. So when we have a group of genomes like this, we can infer a phylogenetic tree that shows how they're related. And what we did in this study, instead of just looking at the bare mutations, just the genetic information, we also tried to get an idea of, for example, what we were missing when we sampled these genomes. At that point in time, for example, there were actually hundreds of genomes that had already been sequenced from Washington State, even though the outbreak there was pretty new and pretty small, and very few genomes available from Wuhan, even though we know that the epidemic started there. And during January and February, the virus was out of control, and probably hundreds of thousands of people were already infected there. So we had this unsampled diversity issue, where we know we're missing a lot of the story. And so how do you deal with that? We also have information from individual cases where a person may have been sampled in the United Kingdom, let's say, but from interviews with that case, we know that they traveled the day before diagnosis from Italy. In that case, that travel history can become very important information. And it kind of gets around this unsampled diversity problem, where if you have very poor sampling from Italy, which we did early in the outbreak, but decent sampling from the UK, these travel cases in the UK actually are a proxy for going in and sampling the virus from Italy itself. And so when we did this, it really clarified the picture pretty nicely here. So here's this outbreak clade in the top right. These are primarily genomes that were sampled in Washington State. We've got WA2, that second genome that was discovered in February, that mic drop kind of moment when the inference was, holy crap, WA2 is so similar to WA1 that we think that WA1 must have directly given rise to the part of the evolutionary family tree that WA2 is found in. And here on this phylogeny that takes into account a variety of kinds of information, including not just genomic information, but also travel history and this unsampled diversity issue, it shows that actually there's really solid information here that WA2 comes from a separate movement of the virus out of China. In this particular phylogeny, it looks like the virus came from Zhejiang state into Washington state, and that there was a separate movement of the virus from Hubei province, where Wuhan is, to account for WA1, and that indeed, despite the only two mutations that separate these lineages, that there was a separate origin of WA2 and its related viruses. If you look over and over and over again at the data in what's called a Bayesian framework, where you end up not with a single tree that you think represents the true history, but a group of trees that kind of together represents the most probable unfolding of that evolutionary history. If you look at that whole group of phylogenies in a Bayesian framework, the evidence is actually strongly pointing to WA2 and the Washington outbreak clade arriving also from Hubei rather than some other part of China. But it's completely possible that it did arrive from some other part of China. What is not really in question is the pattern that this virus had a separate introduction from China. And so what does that mean? Well, it's good news in a way, because it means that all of those heroic efforts that went into trying to prevent transmission of WA1's virus onto other people in Washington state, it probably actually did work. And we thought this was an important finding because I think if this case had gone down in history as WA1 initiating that outbreak, it really would have called into question all of these efforts that public health authorities were trying to get people to do to engage with contact tracing and give people the names and contact details of people they may have infected. Cooperation of patients to actually isolate themselves, and all of this, and so this was quite good news. The other interesting thing is if you look at when this event happened, these phylogenies can also give us some indication of that. And so here we're going to home in on this branch right here. And at some point along this branch, we think the virus moved from China to North America. And look where that is. It's in February. So why is that important? Well, on February 2nd, the Trump administration's so-called China travel ban went into effect. And from that point on, no Chinese nationals could travel from China to the US. And so how does a virus get from China to the US, if this is really the time frame that it happened, if there's a ban on travel from China? Well, the ban was about as tight as a sieve in terms of preventing people from traveling, because US passport holders and visa holders could still travel from China after February 2nd. About 40,000 people flew from China into the US during the month of February after the travel ban. And there was pretty notoriously lax screening at airports and other ports of entry for people who had traveled from China at that time point. So it's very plausible that, and this is kind of what I think is Darwin would have maybe enjoyed about this kind of work, where it seems somewhat esoteric, but at the end of the day, what we're doing here is rewriting history and going from a situation where this Chinese patient WA1 was going to go down in history as the sort of patient zero of the US COVID-19 outbreak. And there's all sorts of potential for stigmatization of the same sort that we saw with gay men during the 80s with HIV/AIDS crisis. And so we're sort of changing the history and complicating things here. And it looks like not only did that particular patient not introduce the virus into North America in a way where it actually got established and took hold, but it may not have even been an Asian person that brought the virus in and started that outbreak. And those actually can be important findings in their own right. So what really was the story? Well, there was early movement of the virus on January 15th, but we don't think it went anywhere. It wasn't until around the beginning of February that we think that lineage got started with a separate movement of the virus from China into Washington state. But meanwhile, right about the same time, we estimate that the virus moved from China to northern Italy, right about the end of January. And it was really in northern Italy that this virus took off like wildfire. It spread from Italy across Europe as a whole, and then moved from Europe to the East Coast, in particular New York City, in mid-February. And that version of that variant of the virus is one that has a mutation that you may have heard of, D614G. This is one amino acid change in the spike protein of the virus that the virus uses to attach to the receptors on the cells it needs to get into. And that mutation probably makes the virus sort of like stickier Velcro. It's better at sticking to our cells and getting into them. And that variant actually then took hold in North America and over a matter of months completely replaced the Washington outbreak clade, which got a head start and at one point was the biggest lineage of the virus in North America, but it's gone now. It's gone. It went extinct and was replaced with this D614G variant. And as I'll talk about in a minute, we're now about to experience something similar where that variant becomes replaced. So once we had these methods in place and answered this question about how the virus got to Italy and North America, my colleagues and I used these same techniques to try to get at this question of when did the actual pandemic in Wuhan start? And there has been a lot of speculation and even some scientific reports like this one on the bottom suggesting that a large fraction of the population in December of 2019 already had antibodies reactive to SARS-CoV-2. Other reports suggesting that the coronavirus was circulating in Italy as early as September. So are those credible? Could that have been the case? And so this is not published yet, but it is available as a preprint. And here what we're doing is really asking a question about how long the fuse is before the outbreak actually kind of explodes into a lineage that you can ascertain and see. And so one possibility in the top left here is that by the time you have enough samples to actually infer an evolutionary tree, that the common ancestor of those samples actually could be the common ancestor of the whole outbreak, in which case it's very straightforward. You just use these same dating techniques that I've talked about already to place a date on that using these gene sequences from later in time, and you're done. You have your date. Unfortunately, that's not necessarily how things work. And in the bottom right, you have a situation where you could sample a whole bunch of viruses from January and February in China, and they might have a common ancestor by this big blue dot here. And you could place a time estimate on that of maybe sometime in December. But you might actually be missing the true index case, the very first person infected with the virus, by days, weeks, maybe even months, because that virus could have been passed along in a very small transmission network that you don't end up sampling much of until this coalescence event in the big blue dot. And to cut to the chase here, what we're finding, and I think this is going to mesh with what we're going to hear from the WHO investigation that is about to report their findings, is it's really, really unlikely that this pandemic started earlier than about October of 2019. So speculation or reports about this coronavirus being present in Italy in September are virtually certain to be incorrect. And we think this will be a kind of important finding for clarifying when the pandemic started and how that process of it moving across the world unfolded. Okay, very quickly now, a little bit about these variants that you've heard in the news. There's the UK variant, B.1.1.7, it's sometimes called, has a whole bunch of mutations, much more mutation accumulated in that genome than you would expect at the pace that the so-called molecular clock of evolution tends to tick in these genomes. And what we think is happening there is that we think that maybe the UK variant and the South African variant and the Brazilian variant, all of these variants with a large number of mutations, including ones that really impact how the virus behaves, may have arisen in rare patients that have a deficiency in their immune system that causes them to not be able to clear the infection like most of us in a week or two. So these people get chronically infected, sometimes for several months. And this UK variant, for example, has a deletion in the spike protein at sites 69 and 70. There's 6 nucleotides that are missing in the UK variant that are present in other variants. And this same mutation has occurred numerous times independently in chronically infected patients who've been infected for a time, built up genetic diversity of the virus, and then have been treated with convalescent plasma. So antibodies from someone with a healthy immune system that puts an evolutionary selective pressure on the virus to come up with a solution. And so we think that this mutation conceivably and others in the UK variant might allow the virus to sidestep that immune selection pressure by similar to the D614G mutant by getting better at sticking to and infecting human cells. And indeed, there's very solid evidence that this, in the UK, this virus looks like it's about 50% more transmissible. When you test patients in their nose or mouth, they tend to have much more virus than previous strains. Again, a reflection that this virus is better at getting into cells, taking them over and making a bunch of copies of itself. And so this is work that has come out very recently, led by Christian Andersen. And what we did here was look at the B.1.1.7, the UK variant genomes that have been sampled across the US, and try to place a time on when the virus arrived. And there's a huge clade in California, and there's a huge clade in Florida. And in both the cases of Florida and California, we think that we estimate that these viruses actually got established in November. So this virus variant wasn't even described in the UK until December. So before it was even known as a thing, this virus apparently had already made it onto US soil and established outbreak transmission chains that are now going very strongly indeed. And we can use these evolutionary techniques to estimate how quickly these viruses are spreading. And here we're looking at the proportion of B.1.1.7. This is the UK variant in the United States compared to other strains. And you can see in mid-December, it was essentially still almost undetectable, but it's now rising steadily. It's rising dramatically quickly in California. It's rising at a slower pace in California. And just to show you what that looks like when you project in time, what we're seeing in Florida with B117 is a doubling roughly every 9 days of the proportion of the outbreak in Florida that the UK outbreak is responsible for. And when you just run that forward in time, we estimate that by March 8th, the scales will tip in Florida, and there'll be more B117 than anything else. It'll be a majority of cases, and within a couple of weeks after that, it'll be the vast majority of cases. And so this is an issue that we want to be very have our eyes wide open about, that this is a virus that in our study, we estimate is about 30 to 40% more transmissible. And that could be on the low side. It could turn out to be just as transmissible relative to other strains as estimated in the UK. And it also, unfortunately, lands more people in the hospital and ends up killing about 1/3 more of the people that it infects. And so the question now is, can we vaccinate enough people before this transition that is on its way to happening, and rescue the population from having yet another surge of fatalities? California is a little slower. The United States as a whole is right in between. And interestingly, if you look at mobility patterns in the different regions, Florida is the highest. California is the lowest of these three, and the United States is right in the middle. And so this might, the speed at which these variants are taking over might be driven largely by just the degree to which people are going about their normal lives versus slowing things down and staying closer to home as part of mitigation measures. Okay, just to finish up here, I want to switch from COVID, and here's this death rate, this astonishingly high death rate in older folks in particular, and not so much at all if you are younger than about 50, with obviously important exceptions. When people have comorbidities, they are at higher risk. But just in terms of the raw numbers, it's astronomically more likely that you'll die from COVID if you're 80 years old compared to 40 years old. But check out what that looked like for influenza in 1918. This is the solid line here. And influenza killed younger people and older people. But in 1918, there was this very unusual bump up in deaths amongst people who were in their 20s, 30s, 40s, peaking right about 28 years of age. And for about 100 years, it was a mystery as to why that was the case. But Darwin has helped us here as well. And this is work that I published several years back in PNAS. If you look at the evolutionary trees of each of the 8 segments that make up the genome of the 1918 Spanish flu, and these are kind of like separate chromosomes that the virus has its genetic material in. You see a very similar pattern in 7 of the 8 genome sequences, where there are descendants of this variant that still circulate today. And they share a common ancestor with the 1918 gene segment, not too far away from 1918. There's also a related lineage in pigs. And again, in all of these variants, in all of these genome segments, except for this one labeled HA, which is hemagglutinin, which is the most important one for our immune response. They're all consistent with an emergence of this genome close to 1918, maybe a few years before 1918. But the hemagglutinin is a very different pattern altogether. It suggests that the pattern suggests that the swine virus evolved directly from the 1918 virus, but the 1918 virus and later descendants actually shared a common ancestor way back early in the 1900s. And this turned out to be a very important inference, because that led me to thinking, okay, what if a virus that was somewhat antigenically related to the 1918 flu had been already circulating in the early 1900s? And the way flu works is the incidence of flu is highest when you're young, when you're 2, 3, 4, 5 years old. So almost all of us get our first infection with influenza when we are that age. And people who had been infected with this variant that may have been somewhat similar to the 1918 flu would have had their first infection with a virus that was quite closely related antigenically. And sure enough, if you look at the pattern of deaths, so this is a more fine-grained depiction of the pattern of death. So this gray line shows you the percent of each age or birth year cohort that died of influenza in 1918. And you can see this very steep peak at 28 years old. The dotted red line shows you the proportion of those birth year cohorts who had been exposed to a strain that was about as different as you could get from the 1918 strain as possible, this H3 strain. And I'll just show you on a phylogeny here. There's a whole bunch of different subtypes of influenza. H3 is up here, and it's very different from the 1918 virus, which is an H1 virus. And so the simple idea is this, that every one of us is exposed to our first flu infection when we're 2, 3, 4, 5, 6 years old, and that forms an imprint for the rest of our lives that is actually extraordinarily good at protecting us if we are later infected with viruses that are somewhat similar to that strain, but leaves us at a deficit if we're challenged by something that's very different than the strain that we first had as kids. And this seems to also explain the protection patterns against these potential pandemic flu viruses like H5N1 and H7N9, where H5N1, for example, if you look in the top right, it almost only kills people born after 1968. So why is that? What is going on there? H7N9, which is in red here, it almost only kills people born prior to 1968. And so if you look at what virus people had been exposed to who were born in these different years, if you were born between 1918 and 1968, the first virus that you had was I'll take you right back to this phylogeny. The first virus that you had was a group 1 virus that was in this blue portion of the hemagglutinin phylogeny. If you were born after 68, chances are the first virus you had was in this red portion and was an H3 virus. So the human virus H1 or H3 are either quite similar to H5 in the case of H1, or quite different than this avian virus H7 in the case of H1. So H1 and H7 are mismatched. And what you see is if you were born at a time when blue viruses were the ones that were circulating and you had as a kid, your chance of dying from a red virus is increased because it was mismatched. You didn't have a red virus as a kid like these people had, and so you have a higher mortality rate. These people born after 1968, they were exposed, most of them, to a red virus when they were kids, and sure enough, they have very low levels of death when exposed later to an avian virus that's in that same red group of the phylogeny. And with that, I just want to wrap up with some thank yous to my lab members, Grace Quirk, Brendan Larson, Tom Watts, all three of whom have been working very hard through this pandemic and doing a lot of lab work prior to being vaccinated, which is very, very courageous. I want to thank co-authors on the work on the evolution of the emergence of the virus in North America. And I want to thank all of you for tuning in for this Darwin Day lecture. And I'm really looking forward to taking questions. Thank you very much.
Thank you very much. The audience can't clap, so I'm clapping for all 264 people that were.
Appreciate it.
Great. So for the audience who's listening to you, please populate the chat with questions. There's a bunch in there now. I'm going to start with a couple of questions that came in during the talk. One is kind of specific to the earlier part of the talk when you were looking at simulations of WA1 case and making the inference that because the common in the Seattle outbreak, the Washington outbreak, everybody was 2 mutations away and nobody had an identical genome or 1 mutation away from WA1. Does your model consider the possibility that those 2 mutations had a selective advantage, and that's why we saw them instead of the 0 or 1 mutations from WA1?
It does. Actually, we looked at whether that might play some role, and to cut a long story short, it doesn't change the overall result, and we've since seen those mutations as individual mutations as well.
I kind of have a couple of people asking kind of similar questions along this line, and that is, once someone's infected, probably for at least most viruses, mutations are happening as this person has a lot of different viruses and variants are forming within the person. And HIV is probably the way most of us are familiar with that happening. Why didn't something like the UK and the South African variant cropping up all the time? And kind of related to that, how do we even know they started in the UK or South Africa? Maybe they started in the US, where we have 25% of the world's cases.
Yeah, so taking the last part first, we don't know for sure that the UK variant started in the UK. What we do know is that all other countries seem to be behind the UK in terms of the timing of the takeover of that variant. So if it did evolve elsewhere, it would have had to evolve somewhere with exceedingly poor genomic surveillance. And so I think it's pretty likely that the UK variant actually did evolve in the UK, probably the same thing for South Africa. And then remind me the first part of the question on the mutations.
Given that when someone gets infected and mutations are always occurring, why don't we see the same UK? If it's more infectious, it probably has an advantage. Should be cropping all the time, right?
Yeah, so that's a very good question. One part of the answer is we did, in a way. We saw this D614G mutant take over in the spring, and it wiped pretty much every other variant that didn't have that mutation that in the lab you can show increases the affinity of the spike protein on the outside of the virus for the ACE2 receptor that it needs to get into. And so the UK variant, in a way, is just the same thing happening again with an even more sticky version of the spike protein. But it is an interesting pattern where there was a long gap between that variant and the UK variant. And so one possibility is just it's a numbers game, and the epidemic really spun out of control in the Northern Hemisphere winter. And there were just many more cases in the Northern Hemisphere winter. Another interesting possibility is it may have had, again, it may be that the crucibles for this kind of evolution are the odd patient that has a poorly functioning immune system that can get chronically infected. And again, there are these cases where chronically infected individuals who have had their virus tracked over time, and the virus seems to be sort of stable, and then you treat them with convalescent plasma, and then all of a sudden you see some of these mutations that we see in the UK variant and so forth. It may have something to do with with convalescent plasma being okayed for use by regulatory authorities. And interestingly, the FDA has just rescinded, so these sort of emergency uses are under what's called an emergency use authorization from the FDA. And the FDA just modified its emergency use authorization for convalescent plasma so that's only allowed to be used on newly infected people. It can no longer be used on chronically infected people. And I think this is a reflection of the concern that maybe it is the combination of lots of lottery tickets with lots of cases, plus more use of convalescent plasma in chronically ill patients that might have resulted in this pattern. The final thing that I'll say is, outside of the UK and a few other countries, we don't really have a great grasp on what other variants are out there and have been out there through this time. And so it's conceivable that we've just missed part of the picture that would fill in the time between D614G and the UK and South African variants.
Okay. I have several kind of questions coming in, getting back to that first index case in Hubei, Wuhan. One is when your model, when you establish the different possibilities of persisting with extinct sibling lineages and then finally exploding, does those consider variation in infectivity rate?
Yeah, we did vary multiple parameters, including the doubling time of the epidemic, which is indirectly related to infectivity rate. And we found that the results were pretty robust to any of the factors that we threw at these models. And that was the case for the German-Italy question as well, that I didn't really talk about. But there again, you had the same pattern where the simulations were indicating that the pattern that was actually observed would be extremely unlikely, not impossible, but unlikely. And then the Bayesian phylogenetics totally kind of independently indicating strong support for separate origins of the Italian outbreak from Germany.
Okay, and kind of more broadly, getting to the origins, a couple of questions. One about over the last, it seems like 10 months, we've heard about coming from bats, from pangolins, bats to pangolins to humans. What's the latest thoughts on which species and how everything started?
Yeah, we still don't know. And I don't think there's any good reason at the moment to incriminate pangolins here. That kind of, there was some indication that a pangolin virus might have had a region that was more closely related to SARS-CoV-2 than bat viruses, but it's clear that pangolin virus came from a bat, and ultimately it's not. There's no doubt a wide variety of variation circulating in bats, horseshoe bats in particular. And so, yeah, it's still not clear that there was an intermediary species, and ultimately it goes back to horseshoe bats.
So related to that too, a couple of days ago, the WHO went through some scenarios of origins, and they considered the possibility of a lab escape, and they concluded that of the various hypotheses they considered, that was the least likely that it was a lab escape, but they could not rule it out. Do you have any sense of, is it a zero probability? Is there some chance, and how likely that might be based on the genetic evidence?
The genetic evidence doesn't really tell you too much about whether a natural bat virus escaped from the lab. It's clearly a natural bat virus. And I would just agree that it's a very low probability scenario. Yeah.
Okay. And I had a question here, which was, how can scientists convey the uncertainty? This is coming from an evolutionary biologist. How can scientists convey the uncertainty inherent in these evolutionary inferences to government officials and to the public without losing these important audiences' trust?
Yeah, that's always a challenge, and you just kind of have to you have to do your best to get down into the weeds deep enough to actually communicate what you've done, and when you do that, part of that is just basic statistics, that we're never proving a hypothesis. We are just rejecting alternatives at particular levels of statistical significance. And that's not necessarily something that people outside of scientific circles are familiar with, but I think it's part of the job of scientists to explain that certainty and that process of fine-tuning what we think happened by rejecting hypotheses at varying levels of confidence.
So if I could push you a little farther on that, I think that's the answer that probably most scientists would give. Presumably over the past 12 months, you yourself have been called on by, I don't know, governor, your state health service, national people. Have you gotten, I don't know, maybe better at talking to them? Or do you feel like, I don't know, are there certain, like, has your personal experience changed on what you just said?
No, it hasn't. And I guess I haven't really been challenged with that explanation problem during this pandemic. And I've done a fair amount. I've had a fair amount of interactions with the press prior to this. And although I'm never great at it, I do try to say things as simply as possible, and that hasn't really changed too much.
Okay. Have any of your models considered horizontal gene transfer or recombination, possibly with other existing coronaviruses that are circulating?
Yeah, there's no evidence for any recombination between SARS-CoV-2 and other coronaviruses since SARS-CoV-2 emerged. That would be very, very easy to spot, and would pop up in analyses very quickly. Recombination within SARS-CoV-2, no doubt, is happening sometimes, but it's hard to see when the viruses are so similar. But I think as time goes on, we'll see cases of very clear cut recombination of this virus.
So here's a question coming in from a student, and I know you, based on your past work with the flu, have an interest in history and tying history into epidemics. So how do you think SARS-CoV-2 will look to future scientists, say 100 years from now, similar to how we are now looking back at past plagues in history?
Well, let's think about it. It's going to look a lot like 1918 in some ways. It certainly had the potential to kill as many people. But I think we're going to have to look back, despite, and it's kind of hard when we're in the middle of the, or at sort of the tail end of the big winter surge where 4,000 people have been dying every day. It's hard to do this, but we're going to look at it as a victory that we did some things that prevented it from spinning so out of control and just rolling over us like the 1918 flu really did. And we're on track to, if we can get some relief from vaccines, to have a very different story in at least some parts of the world. And the challenge will be to have that escape hatch of vaccination offered to people in less rich countries as well, so that we can extend that across the board. At the same time, in the US, you can't get away from the fact that you've got 25% of the deaths and 5% of the population. That's not going to be forgotten, and it's been a major destabilizing event in this country.
So maybe kind of tying into some of your previous work with the influenza, just a couple of days ago, it was reported that there are very, very low cases this year of influenza in the US, presumably because of social distancing and mask wearing. Do you think that is going to alter which of the existing flu strains that are predominant, sort of going forward into the next decade and beyond?
It's a very interesting question, and just one thing that I would quickly say is the sort of very low levels of flu are an indication of that victory that I was mentioning in the previous answer, that if we had allowed things to proceed in a way where we had a normal kind of level of flu transmission, just imagine how many people would have died of COVID-19. It's just amazing to think about. But yeah, I've got a big flu grant that I'll have to think very carefully about whether we're going to be able to do some of the things that we plan to do. But I think what will happen is that life will get back to semi-normal here, and flu will come roaring back to levels that we were used to.
It won't go extinct.
Geez, I think there's very, very little chance that it will completely go extinct, just because of how it can get passed in between different regions of the world, and we're not completely ceasing to move people around the world. So no, I doubt that it will go extinct.
Here's a question kind of related to your influenza work that you mentioned toward the end, where depending on what year you're born in predicts your first original sin. is that going to happen with COVID?
It's a concern for sure that when people talk about just updating vaccines with a new variant, I do wonder to what degree an updated vaccine will serve to just wake up the recall response to the first antigens that you experienced either through natural infection or vaccination. But yeah, I suspect that there will be some degree of original antigenic sin.
I apologize if we have some frozen audio visual. I'm not sure if you can hear the speaker. Yeah, I was about to wrap up the last question anyway, because we decided that 8:20 was going to be the last question. I think we have lost our speaker because of technical issues anyway, with only 1 more minute left. So thank you everyone for coming to the talk. I appreciate it. Thank you for all the questions, even if we couldn't get through all of them. And look forward to next year. Keep your eyesight on Duquesne Darwin Day talk, and we'll have yet another exciting talk, possibly virtual, possibly in person. So thank you once again. Have a good night.